for building the planktoscope.
Yeah, that's tonight.
Oh, is that today?
That's tonight, yeah, at 6. If anyone wants to join, we'll be ordering food.
Perfect.
We can build a planktoscope, so like from 6 to 10, we're just gonna see how far we can get. Yeah, and you can also like participate as much as you want, like you can come and hang out and see how it's going.
Yeah, and I think I cannot recommend more strongly. Really, the sets of skills we're trying to impart are just all based on folks trying things out. And so effectively, to make the best use of most of these sessions, you really have to take the time and actually do the things. There was a thread that I didn't set up as an assignment, which was around the 3D printing sets of parts and Fusion 360 itself. I am just assuming that most of you got a chance to actually try it. If you don't try that with your own hands, there's not much that can be done. If you need help, once you get stuck, there are tons of people, especially Ian as a TA in the lab, who can really help on getting you unstuck. But give that a try. And then in that same channel, you can try on Discord posting some of the things that you've actually done. I'll set up some kind of an assignment around that at some point of time if people need that incentive. But otherwise, we should just get started. And I think today, since I'm doing the lecture online, we will try carving out some time for just discussions and conversations around projects a little bit and the logistics. This would be the week that we will populate the idea board that I'd mentioned. So I think, yeah, let's probably the last 10, 15 minutes of this lecture, let's dedicate to actually thinking about that. Any questions on the chat on paper before we get started? I am curious if anybody figured out the chirality puzzle that I had said. Did anybody repeat that experiment on hanging a piece of paper, wet piece of paper, and letting it dry? I'll show that picture just one more time. Just so that people are aware of what I'm referring to. And I'm curious if anybody did this experiment. So let me share my screen. So this was the proposed experiment. You take a strip of paper, you make it wet, and then you hang it with just a little paper clip. I'm curious if anybody did that. I know Deborah and a couple people from a few years ago know the answer to this. But did anybody get a chance to do it in person? I'll wait for a hand or... So again, if it's not clear, I'll just repeat. It's a very simple experiment. You take a strip of paper, you make it wet, and you just hang it, and you let it dry. And the conjecture here is, and what you can see here, the photograph of re-wetted and dried newsprint strips. So if you do this with newspaper, try doing it with other paper, but definitely do it with the newspaper. The idea is that it will twist. And the question is why? So I think since folks have not done the experiment yet, I'm not going to discuss this. Try doing this experiment, try it with your own hands, see. I'm curious, before doing the experiment, does anybody have any idea? I mean, it's very clear from this image that it's going to twist. Does anybody have any ideas before you do the experiment? Why should paper, when it's dried, twist? You know, wrong answers are perfectly okay as well.
Maybe when it dries, it shrinks. So some parts shrink more than others, making it twist.
Mhm. Yeah, I think you are on the right track, that in something changes in paper when you make it wet and you dry. And that has to do with fibers, the fibers swell. We discussed in the past that the junctions become loose. And then when it dries, clearly there is a volume change associated with it. But volume change alone should not be able to answer. There are lots of materials like sponge, for example, that we all use in our kitchens, does shrink when it dries, but does not twist. But I think,
is there any difference between like any piece of newspaper with prints on it?
no, I think the conjecture is that this should work with literally any piece of newspaper and lots of different types of paper, but to a different degree. So we know something to do with the materials that are used to making this paper.
There's
And I
two
think,
responses in the chat.
oh, let me see. I don't know how to see the chat screen.
Maybe it dries like with a different speed from the left side and right side.
Yeah, that would be one place to think about it, but we are not preferentially making it dry differently. We don't allow, we let what evaporate equally. There's no asymmetry that we are creating. So again, the intuition for these ideas only comes when you actually do the experiment. So I'm waiting for somebody who really cracks this. Try doing this with different width of the strips of paper. That'll give you another hint itself. It really is quite magical when that works. And I'm going to leave that as a mystery, but I think I can see folks have not played with this enough for it to be resolved in some sense. Okay, so let's jump into what I want to do today. I want to jump into diagnostics a little more in detail and talk about the role of paper in diagnostics and other threads. But at that same time, I think interrupt me anytime. These sets of sessions are really supposed to be discussions per se, because you can find a lot of this material while reading itself as well. Okay, so I think we are going to continue where we left last time. And then I think I just want to remind folks, I finally looked up and found those sets of the paper airplane comment that was made. So I think the data essentially is a 2-inch paper airplane survived Mach 7 speeds and temperatures reported to be 200 C in a hypersonic wind tunnel for 10 seconds. I think this is special paper that they put together. It still is classically designed as paper. This was the Japanese experiments that we were talking about. So I think I find it quite interesting in some sense that many of the materials that we'll talk about, especially lignin in plants, have properties that are thermoplastic-like. And those thermoplastic properties can essentially make extremely durable materials that can also survive really high heats. Of course, one of the threads would be, it would be valuable to see what the composition of these sets of papers was. Because when you're thinking about thermally insulating materials, the kinds of materials that are used currently are not that sustainable. Mica is one example, and mica mining has a lot of issues. And a couple of years ago, mica mining was a big project in the class itself. And it would be valuable to look for more sustainable materials that are also degradable, but can survive extreme heat and can still be insulating, which is an important application in all of electronics, for example. So yeah, if somebody wants to think about that. And then another very important material that we worked on during COVID, which really is paper saves lives quite directly, is actually this image in the center. It's called a PAL filter, PAL for the person who invented it. And you can see the combination of filtration and origami in this object. We have thousands, actually tens of thousands of these filters in the lab currently. Because of a very strange incidence during COVID, these filters go inside a ventilator. And one of the challenges with the ventilator is this notion that you want to make sure that if a person is put on a ventilator, you don't contaminate the ventilator itself with the virus that the person has. And in a hospital setting, so it's incredibly important before a person is put on, a new person is put on a ventilator, that all the filter sets are changed and you don't have to clean the machine from the inside. You can imagine that can be an extremely difficult task, because if viruses get into all kinds of surfaces, so then you are passing viruses from a sick person to a healthy person. And the only barrier between the two is essentially these things called ventilator filters. You can see it's a corrugated surface. It does literally feel like paper. I have a lot of them. And very much like some of you know the challenge that happened in the Apollo 13 mission, where they had a square filter that had to fit in the round hole, or a round filter that had to fit in the square hole. Very similarly, a challenge happened during COVID, that PAL is the global supplier for these filter sets, and they ran out of these filters pretty quickly. So there were tons of ventilators that were available, but could not be used because hospitals did not have these filters, and it's unethical to put a patient on another one that is contaminated. And the challenge, so Paul reached out to us and we worked on this challenge together for a while. And the challenge is quite complex, actually, which is associated with, they had the machines that can make the paper cartridges, but they did not have anything that could make a general purpose modular seam around it. So if you think about it, if you're going to make a paper into a filter set, you have to think quite a lot about no airflow should be around it. And then the other reason, I don't know if all of you can see the corrugation or the folds on the left. I'm curious, does anybody want to venture out? Why is this filter folded? Why would you need an origami pattern to fold your filters? Say, for example, in a context of, COVID or in general. Any ideas on why might origami be a useful trick?
To increase the surface area?
Exactly. That's a pretty straightforward one. You increase the surface area, and the higher you increase the surface area, you can use higher and higher barrier in a filter. So the cross pressure or the resistance of this filter, as it gets better and better, gets higher, right? That makes intuitive sense. If you just have a mesh that's tighter and tighter to catch all kinds of things, you would need to breathe more heavily. And so to avoid that as a fact for a patient to breathe more heavily, you really increase massively the area. So inside this little box is actually quite a large piece of paper that's folded in 2 dimensions in a really clever origami pattern. And that's really the magic of many of these filters per se in a medical context. So since many of you are thinking about origami patterns this week, literally these sets of patterns save lives. And then finally, one thread that I just want to mention as a closure is people should take a look at Chairigami. Chairigami was a project started by a Stanford alum some time ago on thinking about real furniture made out of cardboard. And I think there is many groups that essentially do something like that. In the context of COVID, there were hospital beds that were largely made out of cardboard. But I think one of the fun things about this space is there is a lot of effort right now in making durable furniture out of cardboard. And effectively, not just for more temporary applications, but also for longer term applications itself. So on the origami side of the story, you can check out some of the projects around chairigami or, cardboard furniture. Any of you who's missing a chair in their dorm room, give it a try. You'll be quite surprised how good and aesthetically pleasing some of these patterns are. And there's a lot of information online. And if somebody does decide to build something larger scale, do bring it to the lab. There's plenty of space to build something large. I think it would qualify for your origami assignment as well. Okay, so that's all I want to say. And then now I want to jump to the more chemical side of the story. I am curious if you guys can see my screen.
Yeah.
So, okay, maybe we'll do it this way. Yeah, I think the thread that we will follow on is starting to think a little bit about diagnostics and what properties are actually both essential for why paper has become such an important material in the context of diagnostics. And I think there's a little bit of a historical thread that I get very excited about sometimes, which goes back to the discovery of litmus paper itself. So litmus paper could be said to be the oldest diagnostic test. And I'm assuming you've all played with litmus test. If you haven't played with litmus test recently, do find it. It is quite magical. And you all know that, a given set of a compound can turn blue or red depending on the pH that it's present in. But the historical context and the word litmus comes really from a compound that's made out of lichens. And so this is literally 1300. And then the credit for this goes to Arnaldo Villancia. And he happened to be an alchemist. So you can see an alchemist discovering a litmus test kind of has the right ringtone to it. But literally what he was doing and what we discovered is he found lichen. A couple of species of lichen do this as a phenomena. And what you would do is you would grind up lichen. And it turns out lichens happen to have this compound, the hydroxyphenozone, which is really the chemical compound that causes this change. I don't actually know what the physiological function for this compound in lichens are. I'm assuming you've all seen lichen or picked up lichen. I think for some folks who might not live in colder climates, and again, lichens require incredibly clean air. So if you've never had the chance, go try picking up some lichen. It is a symbiosis between 3 different species. And very recently, until, I think I would say 5-10 years ago, we had always assumed that lichen was symbiosis between 2 species. It's only very recently that we have discovered that there is actually the 3rd entity, so algae, fungus, and bacteria, which is just remarkable by itself. But what I'm curious about is, and I didn't realize this, is I don't actually know why lichens produce this compound. And so one side project for somebody who wants to do is see if you can repeat Arnaldo's experiment. Go find a whole bunch of different lichen. There's plenty of it around on campus. Grind it up. See if you can actually make that blue powder. He describes this as a blue powder that when dissolved and when you change pH actually leads to the color change. And I would be very curious if somebody in the class actually repeats that experiment. There are two experiments that I would love for folks to essentially repeat on this front. This is one of them. And, you know, I think, of course, at that time, as an alchemist, he had no idea why something like this was working. But that did lead to a very general test. And of course, until today, we don't make this compound using lichens anymore. But that general principle of colorimetric tests is very broad. And in fact, most of you who have at Stanford taken the color test, which is this COVID test, the LAMP test, which we will talk about in one of the labs, it is also a colorimetric test. And many of those colorimetric indicators are actually pH indicators. It's just that the amplification of a nucleic acid can produce a lot of hydrogen ions, changing the pH of the solution, and hence that can be detected as a color. And some of the sets of tests directly allow readers to actually use color. But literally, the history of this subject goes all the way back to 1300s. Then the other thread, which is historical in some context, is a Russian scientist who then pushed on the use of paper in the context of separation. And then this is really the discovery of paper chromatography. And paper chromatography is not so different from when you paint. Sometimes you might have noticed, and we can do this experiment literally right now. Maybe I'll try to do it, where you will see that the ink that bleeds on paper, when you touch it, the farther it goes, it actually changes color. So I don't know how many of you have noticed that if some folks use watercolor, or you try to use a sketch pen and you leave it on a paper at a point for too long, you would see that many colors appear along the way. So I'm curious, has anybody seen that phenomena? Do people know what I'm talking about? So this is literally separation of chemical compounds, because these sets of chemical compounds are traveling through a matrix, and their associated interaction with this matrix might be different chemically. And if that is the case, you can essentially separate proteins, chemicals that have different chemical affinity, for example. And this really became the first fundamental method that was used for protein separation that was very widely adopted. And again, I think the second experiment, and much of this work, was really on plants and thinking about separation of chlorophyll and different types of chlorophyll. So chlorophyll, many of you might have heard, comes in two forms, chlorophyll A and chlorophyll B. And it's actually different plants will use a different stoichiometry. And it's fairly easy to take a leaf, grind it up, put that in, and literally have many bands of chlorophyll show. And so this would be kind of a second experiment if somebody is interested. The picture on the left is one example of something like that, is you can literally take a leaf, grind that up, and separate out different parts of chlorophyll just in your dorm room. There are a couple of tricks on thinking about how to use solvents for something like this. And then I think one thing we might try to do is on the diagnostics assay day in the lab, we'll try to recreate this as a demo. But anybody else who wants to actually do this beforehand. And then the general principle here is again the same. And if you were to change and treat the paper with different sets of compounds, you could actually generate a separation matrix for different types of chemicals, for example. You can think about the value of this. And I think one problem suggestion that I want to give, which will become relevant in this context. If somebody is interested in the class in detecting fake drugs, I don't think we have talked about therapeutics as much in the class, but maybe I'll set up the kind of the premise for this, is it turns out a significant portion of drugs that are accessible widely globally can actually be fake. And of course, you can realize the horror of a person who is essentially under a medical treatment and realizes that the drug that they have been given is actually has no active compound. And that is disastrous in many ways. And of course, many of the chemical analyses that are used to detect and verify many drug manufacturers are very expensive. And so it ends up being that many manufacturers fly under the radar. And of course, there are bad actors in this space. And this is also why, if some of you have seen that the drug packages have holograms on them or have certain sets of printing techniques that are slightly harder to replicate. But there is a beautiful project that I'm trying to now remember, and it would be fantastic if some of you actually replicated and built upon that work. Does anybody in this room know which project I'm talking about? I'm going to do a quick search to see if I can find okay, so here is one example. Use of thin layer chromatography to detect counterfeit drugs. Is chromatography the key to combating counterfeit drugs? And I'm just gonna find this paper. It's called VeriPad. That's actually built on another work. So just to kind of give you a context of this, governments and health organizations, the counterfeit drug detection device market is estimated to be 1 billion in 2016. So it's a huge problem globally. And so here is another example of thin layer chromatography for detection for fake drugs for malaria. And I think the project that I am after is, I want to show you guys an image associated with this, which is quite striking, and it is not this. There it is. So this is the image I wanted to show you guys. And literally, this is from University of Notre Dame. It has a catchy name. Maria Lieberman. And I met Maria a couple of years ago, and at some point of time, if people get interested, we can also reach out to her. But the image that you're essentially looking at is a set of a paper strip. And what she did is, you can see each one of them actually has, so clearly this is not either a pH indicator or chromatography, but it works on the principle of being able to create a colorimetric signature for complex sets of compounds. So if you're going to apply an idea like this to drugs, you have to realize that drugs have many active compounds. And you would like to know, so say if you're making a general purpose card, you can have a combinatorial reaction that for many active compounds would combinatorially react, which would be a colorimetric output. And then from the signature and the intensity of these sets of colors, you could identify what was, not very precisely, but at least as a baseline first, whether any of the active compound that was being proposed is even actually present in the device itself. And I think what I would love for some of the folks to do is to read a few of these sets of papers. So this is what that card actually looks like. They also show exactly how to recreate this card. They talk a little bit about how to actually make this, and we will talk a little bit about making paper diagnostics oriented things using wax printers. We have a couple of wax printers accessible. You can see the scratch of the active compound that's been added. And this is essentially, think of it as a very low cost combinatorial chemistry kit that's accessible. And even a pharmacist anywhere in the world could pull out this card, scratch the new lot that just arrived, and test whether what he's selling to people is actually real or not. And again, this, for folks that are excited in this space, this is fairly straightforward to print different kinds of, it'll give you a sense of thinking about how to add much more complexity to the kinds of diagnostic tests that are, that you're often used to seeing. And this picture reminded me it would actually be really valuable to explore the space further, primarily because any sets of advances on this, both, of course, it's easy to contribute, but again, any sets of advances in this have a huge impact, especially when we're thinking about antibiotics and, of course, drugs for infectious diseases. Any questions on that? And I think one thread we'll do is I'll post a couple of papers on PAD to begin with, so that we have a set of reaction assays that we might try to recreate in the lab. But I'm just curious if just the general principle makes sense to people. I wasn't planning to talk about this, but it just occurred to me that it's nowhere in my slide deck and should be. And then, of course, one thread in that context that would actually be valuable is for some of the folks who are in places where there have been previous reports of fake drugs. It's actually useful to just go to the pharmacist and also get different types of compounds to actually play with, in the context of, just say, common malaria drugs could be a starting point. So whenever I'm traveling, I do buy a collection of malaria drugs that I can find to then go back and look. And it's actually just a common practice to do in some sense, because we know some of the global cohort connected are in places where malaria is very common. I would actually be very excited if some of you folks tried and at least created a library of the same sets of drug, but from different places in the market. And then exploring whether, how common is this as a problem. Okay, so if there are no questions on this, I'm going to dive in and walk through kind of some of the diagnostics criterias, and then effectively maybe come back to the physics of this in a little bit again. Unless there was a question.
There's 2 questions in the chat.
Yeah, can you read it? I don't see the chat often when I am on display.
Yeah, 1 second.
Chat.
Somebody asks, how does one get the colorimetric output? Is it like different chemicals embedded in the paper? And then Prabhavi asks, can we apply this technique to check substandard antibiotics?
Yeah, I think the answer to both is true. And so one of the threads on this, the colorimetric output, is just a broad assay of reacting agents that are actually already embedded in the paper. That's the cardstock of the paper. And reaction chemistries with these is what's giving you a color output. And now what's actually interesting about it is that that color output is not only measuring whether that active, it's very specific to a given active compound. So you have the capacity to make a very specific card, or you can really think about generally a large, wide range of a colorimetric assay and just test a given set of a drug and say that the composition that is accurate will have this signature, not even knowing sometimes which sets of reactions are directly happening. So you can also discover the correct signature, the correct optical signature, for example, or you can rationally design it. And I think most of the pad threads are rationally designed. And this is also true for antibiotics. And I think with the substandard side of the antibiotics, one of the complexity, because of course, to avoid antibiotic resistance, combinatorial therapies are often given. This is also true for malaria, for example. So when you take a malaria pill, it is not a single compound. It's very rare that we use these mass drugs to be single compounds. For all kinds of pharmacokinetics reasons, there are multiple compounds to begin with. So you might find in a drug, the most common and the most widely available compound to be abundant, but instead of something that was quite expensive, it's not added at all or added in a low enough dosage that it doesn't actually has the same pharmacological effect. So what's nice about this space to explore is not only can you see absence and presence, you could also just understand whether the stoichiometry is correct. And then I think there's a lot of really beautiful papers from Maria's lab. And what I would be excited about is for folks, if somebody wants to expand this space or even think about spaces that something like this would not have been applied to as yet. And then, of course, one of the key challenges in this space always is, moving from qualitative detection to quantitative detection. And so, of course, with machine learning being so valuable and so straightforward to implement on something like this, you can imagine an optical approach to reading this out by taking a picture of this card so that the pharmacist doesn't need to know what the pattern should look like, and the patterns are just predicted by the algorithms itself. I mean, that by itself would be a fabulous class project and would move this needle forward significantly. Okay, so I'm seeing a comment on drinking water quality. I think that's a little bit hard because the pathogens that you're after in the drinking water is significantly lower in concentration. The reason this trick works is because the drugs are highly concentrated compounds that you already are, actively using. And so in that sense, I think there are other tricks that we can think about in the context of water quality that, of course, have to do with chemical mixtures in water qualities. But if it turns down to be pathogens, for example, cholera, bacterial pathogens, Giardia, many of those are much harder to detect because they are in very low concentration. But if it turns out to be chemical contaminants in drinking water, this would be something that's very valuable. And then I hear something on the front of RNA biodiversity. I think that's a clever idea. I think the readouts, again, on the molecular side would not be so abundant. You would have to think a different way of amplifying it. But in general, paper chromatography is a beautiful space. I think another COVID project that we did that was tangentially related to this is to do isothermal amplifications in gel media. So instead of doing isothermal amplifications in liquids, where you know that all compounds can easily mix and diffuse, we actually work with another team at Caltech on doing spot-based detection of nucleic acids. So we could take advantage of the fact that there is directly a media in which the amplicons that are being made don't just diffuse away. And so we could detect them in higher and higher concentration from single spots. And one of the techniques that this Caltech group used was to show that you can detect single molecules of, say, RNA in a gel if the amplification is happening in the gel, because these molecules don't run away too quickly and don't diffuse the signal. So you can get a signal to be actually at a very single spot. So it was called spot lamp. And then maybe when I talk about lamp and isothermal assays in another one of the sessions, I'll bring that up. So RNA, it is true that you could do something like that to combine chromatography with chemistry and reactions. And again, you can see the name of the game here is there is transport, there is separation, and there is reactions. And each one of these three things can be combined in a very complex sets of ways to create new function. Okay, so let me tell you guys a little bit about the sets of papers and things and why paper is actually a good substrate for something like this. So the first thing that you will hear about, and we have plenty of this in the lab, and the reason I'm showing this is I want to show you that price tag. This is called Whatman paper, and these are Whatman cards, and then another entity, it's called FTA cards. These are the most common kinds of places that are used for DNA storage. And one of the challenges is that it's impregnated with certain sets of compounds that can both promote cell lysis and then can also trap nucleic acids. And so if you think about sometimes for surveillance purposes, people would take what is called a blood spot. So for malaria, it's very common where of course, we worked on Inkwell and I showed you guys how to make smears. Often what's done in the field is a third sample is taken rather than taking venipuncture blood, which then has to be immediately transported to some storage facility. A drop of blood is added to a piece of paper. And the paper has lysis buffers and has, proprietary sets of compounds for capturing DNA. And the reason I'm kind of smiling and smirking a little bit is that you can see the price tag of that. So, 25 pack of these, which will be around 100 samples, is roughly around $100. So that's $1 per storage. If I want to run a malaria surveillance program for genotyping, because one thing you can actually do is you can use these blood spots for sequencing. So if you wanted to run a program like we ran for COVID, for example, for genomic diversity of malaria parasites that are circulating, that would add $1 per blood spot for every single test to begin with, which becomes really prohibitive. And so one of the threads is to really reverse engineer and really think hard about, okay, what is it that you actually need to make a type of a paper like this that can be used in surveillance and other sets of scenarios? And actually, the irony of all of this is just you have to go back to literature. And the first paper I could find that really talks about this idea of interaction of paper, which is cellulose, and molecular compounds, say, for example, DNA, is really this paper from, I think this is 1960 and 1978. And I'll kind of read that in this context of what it really talks about is an interaction or creation of a DNA cellulose bond. And so effectively, the reason paper is actually a very good DNA capture material is because DNA essentially directly reacts with cellulose to form these sets of bonds. And then many of the very much like, this is around the time that people were just starting to work and find materials to handle DNA. And what's incredible about this paper is, for the longest time, this paper was just hidden and somebody at Princeton decided to do this experiment. They showed you can capture DNA with paper and nobody paid attention to it. And it just sat there for literally 30, 40 years until somebody brought up its applications in a diagnostics context. So this is also why I often emphasize this idea that you should all read literature very broadly. And one of the things that they were able to show is actually DNA is an incredible compound in the context of this set of a binding. And then again, I think this is really the reaction that they discovered. And as you can see, essentially, this is one of the threads of the cellulose, and you can think about this as in this fibrous format. And then in these chains of cellulose, essentially, you get DNA binding. And once that happens, you can essentially capture this material. So now imagine if you have a lot of molecules and threads in a given cell or in a given biological component, you really have to separate out the nucleic acids before you can start any amplification processes. Because there are lots of proteins and compounds in biological samples that will inhibit any amplification reactions. And, much of this discovery was before even the discovery of PCR. So people knew how to capture DNA, but didn't know what to do with it because you're stuck with having to amplify it. But now you can see if you can then combine this with amplification, that gives you a remarkable way of then detecting. And all of this is literally on a single piece of paper. Any paper that you have, there is a very nice clear chemistry that you can do. And so many types of papers have been demonstrated. And I think the irony of all of this is this was the demonstration around the 1960s and 70s. And finally, really the paper that demonstrated the value of this idea in a context of diagnostics and for genomic collection is literally all the way in 2017. And this is a remarkable paper that really talks about nucleic acid purification from plants, animals, and microbes in under 30 seconds. And so for some of you who were working in the context of COVID, or at least were paying attention in the diagnostics, one of the key challenges that we ran out of was extraction buffers. So extraction of DNA and RNA was a huge bottleneck in many of the sets of diagnostic tests that were done. And I just want to spend some time on this protocol because it's so simple. If you are looking to extract genomic material from, say, a plant, the first step is you shake and lyse the tissue. So you take a plant and you put 2 ball bearings in it. I don't know if you can actually see this, but those ball bearings are right there. You see these 2 little metal balls and you shake it. And what that does is the metal balls rattle with each other and they mechanically lyse cells. It couldn't be simpler than that. Once that has happened, it says dip for 3 seconds to bind nucleic acids. Then the next step is dip into a wash buffer for 3 seconds, and then dip into the amplification reaction, and then you just run your PCR. So 3 dips, and you can count this total number of seconds. It's pretty hard to do it so quickly, but maybe around 15 seconds is a fair. And so in that context, they show that, okay, 30 seconds is good enough. And the method is very general, because again, you're just after nucleic acid. Again, going back in the context of frugal science, performance versus cost, it's not the most efficient. You're not going to get all the nucleic acids out. So if you have samples that have really small amount of nucleic acids, then you might be better off doing many of these other magnetic-based separation assays. But if you have abundant nucleic acids to begin with, which is majority of the times in many of the diagnostics cases, for example, then you can really utilize a tool and a probe like this. And again, I think, one of the factors of things like these are just the simplicity of it, but the irony, which is, again, I want you to realize that anybody could have done this literally from the time in 1970 when the first proposal was made that DNA binds to cellulose. But it's just the right set of people. They were, for the lack of a better word, basic scientists who just wanted to understand DNA reactions. While the person that was needed to come up with this idea needs to be somebody who's looking for simpler methods. And I think, this is sort of the purpose of this class. So what I'm excited about is who amongst you is going to find that hidden 1960s gem that open up a new application. And again, this is why you all need to read old papers. It just, it's something that's so much incredible knowledge. It is hidden, and sometimes we don't actually exploit it or utilize it in a manner that we should actually utilize it. Okay, any comments on this? And then I'm going to jump to sort of breaking down some diagnostic devices for you guys. Okay, so since I don't hear any questions, I'm gonna show you probably the most famous patent that you should all have read in the context of COVID, but in general, which is the original patent for the lateral flow immunochromatography assay. So, we have used RDT as a word all this time. One of the threads that you will see is, of course, some of you can go and read in the context of thinking about just generally how a pregnancy test works, or for example, how a COVID test might work. And you can think about this in the context that, again, as I said, there is transport, which is you have to get the material to the right place. There is reaction, which is you have to capture something. And then at that same time, there is readout. And the simplest way that this works is you can see on the left side, you have, say, a sample like urine, and that sample can have, different kinds of sets of compounds that you're interested in essentially detecting. And then there is these two test strips. And you can see these sets of test strips are essentially, in this context, happen to be immobilized antibodies. These would be the test zones. You've all seen this test zone. One is a control line. The other one is the antibody that you actually care about. And then the other part of this scenario is there is a wicking zone here, which essentially ensures that all of the fluid that you put in will actually keep traveling. So often enough, if you think about this, the wick zone, the way to think about it is that there is a massive amount of liquid storage that can happen in the wick zone to make sure that the total, all the sample that you put in actually transports and goes from left to right. And then, of course, in the context of thinking about sets of detections, there are all kinds of clever tricks that are used. So say, for example, gold nanoparticles or other sets of particles that will essentially illuminate and make your test strips light up. Because if just an antigen and an antibody bind, you can't actually see it. But one of the tricks that's played is you can have essentially nanoparticles that enhance and give you a visual context that you could literally see it visually. But one of the fun things about this is, this is kind of a core of all of the technologies. We will spend a time in the lab to open up some COVID tests. So if folks want to bring at the lab session, maybe this week, Friday, try bringing as many different types of COVID tests that you might find. We won't run the tests, but we will break them down and we will try to kind of identify what they're made out of. But it is actually worthwhile going back and reading the basic test or the basic patent really is a conceptualization of this idea. It's incredibly simple. And I think its simplicity is what drives its functionality in some sense, because there are no valves, there are no pumps. The pump is paper itself. And actually the patent is fairly old, as you can see. So this is from 2002. Patents usually have a lifetime of 20 years. So it's a little bit ironic that the 20 years are passing just around now. I hadn't realized that literally the LFA's patent has just expired, which is interesting because when patents expire, wide adoption of those technologies become possible, or at least there is wider adoption, if not completely widely available. And one of the things that has been done, for example, is a couple of groups have tried to make a general purpose kit for anybody to prototype their own RDT assays itself. And so I think there's a couple of papers out there that really talk about what papers you should actually use, what should you make WIC out of, how do you get any of the active compounds that you might be interested to be stable. And one of the big bottlenecks in this scenario is really the discovery of the antigens and the antibodies associated with what is that you are after. Like, for example, when I told you all about the HRP23 gene deletion, it was associated by the wrong choice of the chemistry to begin with. And then it takes a huge amount of time currently to really do animal testing to be able to discover and find the most reactive capture probes. So this is the huge bottleneck. And this is why, although you can make the mechanics of an RDT within half an hour in your space, there are robots out there that can literally print RDTs. The big bottleneck of why it takes so long to create quality RDTs, many a times, literally, sometimes even 5-10 years, is the fact that it's an extremely laborious process to identify the conjugate part. And again, lots of machine learning innovations are currently happening in that space, including a couple of people in our department that are working on discovery-based approaches just purely from a sequence space. But that happens to be the key Achilles heel in the development of lateral flow tests. And I think, there might be a few sets of changes coming, but currently that happens to be one of the big challenge. So, and I think I realize some of you might have missed the malaria section, especially the global class, but I had spent a lot of time, and I just want to reiterate this a little bit, that we are at a really important crossroads for growth of thinking about new tests for malaria, and especially in the RDT space, primarily because the two genes that are currently used in these sets of RDTs, the PFHRP2 and the PHFRP3, there is very broad range of gene deletion in sets of places where this has been tested. So I think it again goes back to that same scenario. Because it takes a long time to develop another test, the next test for RDTs that were detected was based on these LFM genes. And I think they are not as sensitive, for example, because just the expression is not high enough. So you don't get enough antibodies in the blood itself. And so I think one of the threads that would be valuable is folks that might want to pick malaria and malaria RDT as a challenge. We'll kind of dive in a little more in this process itself. This would be a phenomenal project for somebody to think about, at least a group of people, because this is going to be a bigger and bigger problem in some sense. And then in case if people want to get a sense for how to build RDTs from scratch, there's no better way. The other space on projects, I'm just mentioning this, that we've been thinking about quite a while, is there is a huge problem with thinking about the faint line. So how many of you have seen what I'm talking about, that faint line on a COVID test? So you all know what I'm talking about. The problem with that faint line is that it can be missed very easily. And what's happening in that process is to just think a little hard about what is it, the process that's amplifying and giving you that optical signal. And one of the things that we have been doing in the lab for some time is implementing optics on RDTs. So this would be another great project if somebody's excited. This week, we will assemble fold scopes in the lab session. And then I think I have to figure out, maybe Ellie and Abina, it just occurred to me, if you could ask all the people in the class to send out an address, we're going to try to ship them fold scopes and a little kit to all of the students that are not physically here. It will take some time, but it just occurred to me, we should have done that at the start of the class. So anybody who's listening and is on Discord, just send out privately to either Ellie or Rabina a shipping address, just so that, I don't think it will reach by this Friday, but eventually we'll try to get you all some fold scopes to make sure that you can, your you've actually played with that tool itself.
Not a Google form, so that way it's not.
Yeah, I think that's perfect. Just do it on a Google form so that the addresses are still private to the class, and then we will. I'll work with Jasmine on figuring out a way to ship out some of the units. Anyway, I think the reason I mentioned that is one of the big things that we have been exploring is this idea of implementing optics on RDTs without increasing the cost. Could you actually amplify the sets of signals that are currently present, even for a faint line? And so using other tricks rather than just your eye to be able to detect the readout of an RDT. So I think if somebody is interested in that as a project, talk to me over this on Friday Lab. Okay, so I think let's dive in a little bit, and maybe I'll share just a little bit in the context of what's been done in the space of paper and diagnostics. This is a massive space. And I think, again, just like origami, the only way you can really gain confidence and a better understanding is to actually try to do something. So, you know, without showing you, for example, how it was done, I'm curious if you now want to do paper diagnostics and microfluidics. This is a very famous picture from Whiteside's lab. It's beautiful and very clever. But if you wanted to have compounds not mix, but still cross over each other, how would you implement that? I mean, everybody can see what this picture is, right? You can see that 4 colors have been added. They traveled along these channels, but ironically, they did not mix. So I'm curious if anybody wants to venture out and propose, if you were to do this, how would you do that? Any ideas? How would you make out of paper transport that literally passes through, but does not mix?
So Smriti has suggested using a hydrophobic layer and then a hydrophilic layer.
Yeah. So I think, yeah, that's the trick. The trick is that it is not a single sheet of paper. And so if some of you who saw that image on the left, it's essentially multiple sheets of paper that are then folded together. And one of the contexts to thinking about is that at the crossover point, the fluid goes down and then crosses over and then comes back. So this is very much like weaving or knitting. You know, when you knit for a cross stitch, you have to go under a thread. So, you know, it's possible to make 3-dimensional fluidic channels. And again, this is much more oriented around increasing the complexity of the kinds of tests. You know, ironically, there has been an incredible amount of academic work that has been done on these sets of ideas. I haven't seen widespread products that actually utilize the sets of complexities. Literally, any LFAs that I have still seen are all single strips. And so one thing to think a little bit about is thinking about the limitations of paper itself, and what you cannot do is an important criteria to think about. But it would be valuable. This is a very large class of body of literature called paper microfluidics. And for folks that are interested, it's fairly straightforward, again, to implement. You can come up with all kinds of clever tricks. And this becomes really valuable when you're thinking about multiplex testing, when you want the test to not just provide one thing, but 5 different things, for example. So, you know, if you're looking for many sets of compounds to be tested in a urine, or many sets of compounds to be tested in water, when it goes beyond one, you can really exploit the sets of ideas associated with the complexity of design. And as a designer, it's fairly straightforward to implement, but there is a beautiful amount of mathematics hidden behind this. And I think for folks that are interested, we can run a little bit of a fluids session associated with it. But the simplest way to think about it is essentially, again, that there is some kind of a differential pressure that's driving the flow, and the flow is driven by the amount of pressure and the resistance associated in this media itself. Except this media is actually a very complex media. And so the types of complex media that are described in that space are described by what is called a Darcy's law. And Darcy's law is essentially the law for flux in disordered media. So when you really think about soil and you pour some water in your plant, which is very dry, you notice that the water disappears very quickly. So it follows the same flux law, except now you have that same set of an idea, but in 2 dimensions. So these fronts can get very complex, for example, and you can get instabilities in these sets of fronts. So sometimes you can see small amount of inhomogeneities can lead to patterns that would be quite surprising. And again, going back to the comment that I had made earlier, what you're trying to do is also make sure that none of the reagent that you want to really pass through is left behind. And that's also why for many of the tests that you actually do, you all might have noticed that you add a certain dilution buffer, and then you add your samples to that dilution buffer and then add it to really make sure that you don't run out of fluid. Was there a question? Okay, I think I'm gonna mute you, Kenyig. Sorry. Okay. Okay, yeah, I think that has to happen in all Zooms at some point of time. So I think, one of the fun threads to think a little bit about this is, again, the physics of it is incredibly beautiful. And at that same time, there is this intuition that you might have in the design. And what would be valuable is if some of you pick this up as a project, is to think a little bit about building on your intuition and then testing it with the theory that's already present and seeing where this varies. And so, for example, one of the common things that you don't know a priori is the porosity of your paper material that you might start with, or its wetting energy to start with. So you might do a certain class of experiments to essentially fit the data to be able to identify those parameters. So, you can measure certain kind of a criteria for a flux, but it's much more difficult to measure. You can measure the viscosity, but it might be much more difficult for you to measure the permeability of any given sheet of paper. And so if this law is true, you can then apply, it's very much like the same thing that you do if you have a resistor and you are trying to identify the resistance of it using the current voltage relationships. But now this resistance is essentially of paper. And then I would be very excited if some of you take this on as a way of testing to write down the permeability of different types of paper. And it'll give you a good context for why nitrocellulose, for example, is used so commonly in all our diagnostic tests. So you can do this as a comparison between the rate of flow of a given, say, buffer solution, say an aqueous buffer solution in a RDT test that you've opened up, and also on your A4 sheet of paper, and actually compare the two. Because it's valuable to just understand what types of parameters that you can change that allow you to be able to tune the properties of flux and flow associated with this. You don't have that much choice over viscosity. Of course, saliva and nasal swab by itself would be too viscous. And so that's why for all COVID tests, you actually have to start with a buffer. And of course, you're trying to dilute them in the buffer for all kinds of other chemical preparation of the sample itself. But the viscosity alone is actually very important, for example. And then I think one of the threads that I want to mention in the last 10 minutes is Actually, yeah, maybe let's talk a little bit about a broader range of technologies and threads that are accessible, and then we will go back and talk about WHO criteria. So, I think we talked quite a lot about lots of advantages associated with paper-based approaches. It is instrument-free, often said, it's biodegradable, it's disposable in many types of contexts. Most of these are single shot type of reactions. And then I think there are certain sets of technical hurdles as well. There are limitations associated with the flow control, how much you can control. It really is a one-shot type of a reaction. If you want to create isolations, or you, for example, do a step like a nucleic acid step where there is a wash buffer, that gets much more complex to actually build. And then again, the readouts are often limited. And then there is lots of places where paper has been implemented in a more complex device, which you will see in the class a little bit later itself. And so to add to that complexity, there is a really beautiful space that's growing, which we haven't as yet talked about in the class, is integration of paper and electronics. This is from a review and a book that a friend of mine from South Africa, Kevin Land, had published. It's called Paper-Based Diagnostics. I'll kind of cite the book here, but it's a really fun book that just puts together all the advances in paper-based diagnostics that have been put together. And I'm just choosing a few highlights of this. But if folks are interested, we can try making this book accessible to people for reading. But one of the first things that I want to show you all is electronics. And so you can see it's fairly straightforward to implement an electronic circuit. There are two clever tricks. One is just screen printing. So how many of you have made a t-shirt by yourself before? Any show of hands? If not, we got to make a t-shirt for the class. Come on. I guess the designers in this room here, let's actually propose and post some Frugal Science t-shirt logos. I'm just surprised, Deborah, that over these so many years, it didn't occur to us to print a t-shirt for the class. I mean,
yeah, let's do it.
yeah, let's put together a common logo, and we're going to try doing screen printing. I realize it's such a valuable technique. So am I hearing this correct, that nobody has done t-shirt screen printing here before? Okay, I don't see any. Oh, okay. So do you, one thread would be is, do you, if you've run it, it would be fun for you to run a session on screen printing for t-shirts. Screen printing is an incredible technique. Now I'm diverging a little bit, but I just had assumed it. There is a mesh that you have, and you have an ink, and you essentially block the mesh by printing on it in a manner that it's only porous in certain areas and not porous in others. And you squeeze paint through it. And essentially there is an absorbing media like a t-shirt on the other end. Now, it's not just useful for making t-shirts, it's actually also used very widely in flexible electronics. So there is a lot of silver-oriented inks, graphite-oriented inks that are present, which can literally be printed on paper just using screen printing. So very simply, very common day-to-day materials, completely manual processes can essentially enable complexity of functionality. For example, RFID tags. Imagine if you want to tag and track every diagnostic test in a manner that's electronically tagged. You can very easily print an RFID tag on it. If you have an electronic detection mechanism, but you don't want to put a battery inside because batteries are problematic for many reasons, you can also put a coil for an inductive charging of that diagnostic test. So there is no literal battery inside the test, but it can still operate using electronics. So there's lots of reasons to include printed electronic functionality. And one of the threads is, this is a fairly simple lab session, so we'll try doing this. The other way to do flexible electronics is also using craft cutters. So these vinyl cutters that are used in poster shops, and I think, yeah, maybe at some point of time, Ellie, let's try finding one of these small, I mean, we have the vinyl cutter in the lab, but we can also get a smaller scale craft cutter, which is essentially a CNC knife, and copper tape by itself is analogous to making printed circuits. You know, you can imagine if you want to add heating functionality in a nucleic acid-based test, you could literally do that by a thermal resistor printed on a piece of paper as a mechanism for amplification. The other side of that story, of course, is, you know, so for this example, this is from the group in South Africa. You can see it's a kind of a business card style paper. There are certain sets of electronic probes here for the readout. This is essentially a potentiostat, and so it changes its resistance as a function of a certain chemical readout, and hence you can have a purely electronic rather than a colorimetric readout of a RDT tile test. And then again, you know, as we were talking about this, this is an orthogonal axis of thinking about how to read that faint line, because you can imagine the faint line could actually be read electronically. The electronic signature or the resistance across that would change as a function of the number of colloidal particles or gold particles that have been captured, for example. So there is a lot that could be said in terms of thinking about the readout associated with it. And then, of course, the other part of that story is in terms of thinking about detectors is implementation of LEDs. So this is another example. And there is a commercial version of this type of a kind of a circuit board that are ultra thin. And I think one of the threads is in a lot of tags that are currently used, these sets of ultra thin printed circuit boards are also used. So at this point, this as a technology has scaled up quite widely. But of course, the fun part of what I was describing is here is the pad, and this is literally the screen print. So you can actually see the threads that make the silk screen, and you can see the template on which the silk screen is being put on. And so there is a very wide range of sort of threads associated by using silk screen as an approach. And of course, t-shirts are printed in a very large scale. So you can imagine making also very large circuits that are harder to make by etching techniques. For example, if you care about a diagnostic test that is very large in area for some reason, because you want to capture a large amount of volume, you might want to rely on techniques that are actually using large areas. And then kind of, I think that the key thing that I want to mention in this set of a criteria is WHO, when they were thinking about this sort of a context of diagnostics, released a criteria which they often think about as a way for what is a good diagnostic test. And that's called a reassured criteria. It's incredible that they could come up with such a meaningful acronym for something like this, but it's a good thing for you to remember. If you are thinking about a diagnostic test, often enough, the types of things that are thought about, again, clearly hitting all of them is difficult. And that's why it's important, but it is important. So one is real time, being able to tell the patient as quickly as possible what they have. The second one is ease of specimen collection. So, thinking about if you are doing a test that requires cerebrospinal fluid or a spinal tap, it can be done, but it's quite difficult. But if you could get the same answer from urine or from, say, a finger prick blood, that would be a much more easier. And again, this is why there was a big shift in many of the COVID contexts from the deep nasal swab to the shallow nasal swab to actually a saliva. because of the ease of specimen collection, because the harder the specimen it is to collect, the more problems that there can be and the less accessible the test is. Of course, affordable is obvious. That's what this class is about. Sensitive and specific is a very important sets of criteria, and this has to do with your false positive and false negative. And I think when we will go down the path of nucleic acid test, we will talk quite a lot about it in terms of what criterias make the test sensitive and specific, and how do you do a trade-off between from a context of cost. As you can see, that nucleic acid tests are much more sensitive and specific as compared to the RDT tests that we are using for COVID, but it doesn't mean that one is better than the other. It is the context that determines what tests you need to use. Because the other tests are not affordable, it ends up being where you have to rely on a certain different test from a perspective of cost. Of course, user-friendly is most often ignored in the past, but if folks are interested in at-home tests, that becomes a very important criteria, because the trend is moving towards reduction in costs by literally engaging the users themselves. And it also gives and democratizes the access to the test itself. But most often, FDA, for example, from an approval process, looks very closely at the usability studies. And these usability studies have to be done in a very broad demographics for you to actually quantitatively prove that your test is user-friendly. Rapid and robust, environmentally friendly, and equipment-free. This is probably the most ignored criteria of a diagnostic test. Most diagnostic tests end in landfills, and the plastics that are used are a huge burden because billions of them are made. And on the other hand, there is a very few materials that have been identified that actually work and operate in this context. And one big project and a discussion in the lab that we have had for a while now is to identify biodegradable materials that are sufficient for an RDT test. So this would be another project if somebody is interested in. I mean, I had already said billions of these tests are conducted every year, and you can imagine where they go. So they are all in our oceans and in our water bodies eventually, after they are gone through some kind of an a process associated with either a heat treatment or places where biologically active compounds are deactivated. Many of them find their ways into our environment. And so I think this would be a huge thread in terms of if certain sets of folks are interested in. And again, you know, Abbott's test, for example, the current BNX now, as you've all seen, has a card-based mechanism. It does not have a lot of plastic. And that is really around reduction in the total plastic used. And then finally, you know, it has to be deliverable. It has to be shippable in some sense. And so this criteria is valuable. And I think for certain sets of projects that focus on diagnostics, this is something that you want to value yourself or evaluate yourself based on how many of these things you can hit. You know, it's not trivial to think about, but on the other hand, it is a meaningful goal. And many sets of technology drivers are associated with this as a fact. And then this applies more broadly to not just the paper-based tests that we were talking about, but in general to a broad range of tests. And I think as you will kind of see, one of the points about this perspective that Kevin had written was really around the idea that paper-based diagnostics do actually meet a large range of these sets of criterias. And I think, you know, to a certain extent, that's true, although there are certain class of limitations. So molecular tests over the recent years have been started to be implemented in these sets of frameworks. And then again, you know, I think I'll post this on the Discord channel itself. There is a lots of, you know, things that can actually be implemented on paper. And so for folks that are excited, this by itself and implementing a large number of these capabilities by itself would be a fun criteria. So I have run out of time, but I want to end with a fun video to show you that you can also have fun with paper. sometimes things that you do with paper can just bring joy to people and not just, so this was a project that became very popular. This was a friend of mine at grad school, and he literally did a PhD on this. And I'm just going to play a video without telling you too much about this. And my hope is, I can't find the originals. It's always fun to find the original, but let's see if I can find.
It's simple as you put a line, you touch it with your finger.
Okay, so I'm just going to play this video. This is Jay. And again, sometimes you don't even realize paper and drawing can make people happy. So I'll let it play and then try figuring out what's going on in the video.
Drawing music. It's simple as you put a line down, you touch it with your finger, and then you start drawing music. Raw audio. Stuff like that.
So I don't know if folks figured out what was going on. Did people figure out what was going on?
Like it
Yeah,
kind
you
of,
can just shout out.
yeah, like it kind of seemed like there was like a almost like a gyroscope or something in the pen. Sometimes it seemed like people were following the pen almost.
Yeah, so I think I should have clarified. What this is a musical instrument made out of a pencil, and the music plays. And again, this is an open source instrument. Any of you can make it. We could actually just do a dedicated lab session on this, but there's plenty of videos around. What was at the tip was actually a very tiny speaker and a small battery. And the object was measuring the resistance. It's clipped on your hand. So it's measuring the total amount of current that's passing through your body, and you're touching one hand. And then, of course, as you all know, a lead pencil is primarily graphite. And graphite is incredibly conductive, and that's what actually led to the discovery of graphene. And what you're actually measuring is the electrical resistance of your drawing. And you can take that resistance and map that to an audio tune. And that's how they were making, for example, a piano by literally drawing a piano. And so kind of, I think, again, you can apply that as an idea and think about, oh, wait a second. Once electrical resistance on a sheet changes, you can quantify it in all kinds of ways, how that might change with chemistry. There's so many things to be done with it, but the first thing to also do with it is also to just play. So if anybody is interested, this would be a fun side project. And again, the documentation for how to make it is pretty trivial. If you've never done electronics, it's a great entry project. It's called Drawdio. I can post some Discord links. I know I've run out of time at this point, but I wanted to make sure that you, as you're thinking about serious applications of paper, try thinking also about fun applications of paper. And sometimes while having fun, you might actually discover something very deep and meaningful, and the other way around. Okay, so I'm gonna close at this point. I already realize it's past 10. I can stay online since I decided to do this on Zoom for a little bit, for 5-10 minutes, if people want to just have a chat, if they have questions, if you might have questions about how we are going to organize projects and others. But on the other hand, for folks that have to leave, we'll call it a day. There's no obligation to stay. But on the other hand, if anybody wanted to catch me, many things you might have ideas and things in your head. We kind of do this as an open mic at the end of the class. So this is kind of the open mic time. Bring up anything you want. Otherwise, I'll see you all in person on Thursday. And of course, unfortunately for the global class, I can't see you guys in person.
There was a question, I think there's some confusion
Yes.
about the for Friday, if you could clarify.
Oh, what's the question?
I think there was just some confusion about what's the assignment due Friday.
Oh, remember, the assignment is from last Friday, which is you're making something clever out of origami, broadly defined. So the assignment hasn't changed. I'm just thinking about future assignments.
Okay.
Yeah. Yeah.
I have it. I just haven't really got it.
Yes, yeah.
Okay.